Thank you. My name is Laurence. I'm a PhD student at the University of Tübingen for the Quantitative Biology Center. And I'm going to talk about CRISPR and more specifically about targeted and screening genomics. If I can switch slides. Yes, I can. Okay, so for the next 10 minutes, I would like to firstly introduce CRISPR-Cas because I think it's quite a neat topic and it's cool science. And I'm also highly aware that a lot of you in the room might not use the pipeline, but Christmas is right around the corner. If you need an icebreaker for dinner or something, or to switch topics, maybe this could be it, and that would already be a win for me. I'm going to talk about the different use cases that we cover in our pipeline. It's a pipeline that is on NF Core, and kind of walk you through the different use case scenarios that a user or a scientist can have. And then I also know that we have developers in the room and more techie people. This is going to be my Sequeira version of a talk. I'm going to talk about the fun quirks that I think our pipelines have, and also the kind of research we want to take, the different directions we want to take. So what is CRISPR-Cas? CRISPR-Cas has evolved from bacteria and archaea, I don't know how to pronounce that one, but from bacteria. It is a defense mechanism against viruses. So basically, it figures out the DNA viruses, it goes in the bacterial genome, and it kicks it out. We were able, the last 20 years, to, instead of giving it the viral DNA, to recognize, to kind of tailor it. So we evolved to really precise genetic modifications within the last 15 years. So here on the little, oh, I think I have a laser. Cool, okay, it's like a cat. So the blob here is the nuclease, which is really the scissor part that will go into the double-stranded DNA of the cell line or the species that you are investigating. And then here, the little loop with the, here the little loop with the hairpin is the single, it's called a single guide RNA, and it's 20 base pair, and you can tailor it and modify it the way you want to. So that is generally quite powerful because it reduces massively off target, so you're not cutting the wrong part of the genome, which was what was going on in the science that was previously used. This led to a Nobel Prize awarded to Emmanuel Charpentier and Jennifer Doudna, who has a startup in Canada. And it is in 2020, and it is the first time that 2 women were awarded the Nobel Prize in science. Luckily enough, no woman got the Nobel Prize in science this year. I guess we had our diversity year 4 years ago. So what can we do with CRISPR-Cas technology? We can knock out a gene, which is what I just described, through a double-strand DNA break. By breaking it, we are triggering the triggering the DNA damage response. So heavy machinery is going to come where the cut has happened, and it messes up, basically. It kind of puts, we have different kind of configuration cases, what can happen. And when it messes up, it silences the gene because we maybe skip a base pair. It's not really, like, it's tied in together, like, very roughly. And then the gene is silenced. However, we can also now insert DNA or entire genes. So this can lead to really tailor and try to figure out different responses that we can do when we are looking at curing diseases. And the same way, we can also now just edit one base. Here we are modifying the Cas9 nuclease or the Cas nuclease to not really break both strands of the DNA, but just put a nick. So I read it up, and it is used, for instance, in sickle cell disease, where we really just modify one base pair. There's also ethical questions, which I found really fascinating. I'm not going to cover them in my talk. I do want to mention that we don't, our pipeline doesn't allow it to analyze anything unethical, but I will mention that there are already some, like one scientist used this technology and the science to modify a human embryo. So there are twins right now. There are around 4 or 5 that were modified with the CRISPR-Cas technology. So this I can do with only one gene, right? I can really modify and silence one gene and look at what's going on. But if I want to cure cancer, I can kind of take my cancer cell line, introduce a library where I'm going to go each gene, knock it out, and if my cell dies, I might have found a cure for cancer. Am I saying that our pipeline can cure cancer? I'm kind of like trying to plug that in, like kind of like a weird flex. It can't, but this is called screenings. We can figure out the essentiality of a gene by knocking it out. If the cell dies, we have more information, and we can do downstream analysis where we can then really target that gene. So this is called functional genomic screening with CRISPR-Cas. And we basically target all of the genome, and we sequence that, and we look at what happens. There's a lot of wet lab experimental design that you have to respect, because one of the questions I had when I started looking into this is like, how can we make sure that only one gene is knocked out in one cell, because this is what you need. So you need a certain amount of cells, you need a library that's a certain size to kind of dilute everything, that when you sequence it, you're sure that you have accurate results. So this is what our pipeline does. I have to admit I had a minor breakdown in my hotel room yesterday because I was practicing this talk and I noticed that in none of my slides did I write the name of the pipeline. So, okay, cool. Now that I got a giggle out of you, the pipeline is called CRISPRsec. It's called crisprseq. It won't appear in the next slides. It's on NF core and it's called crisprseq. So like CRISPR and sec like sequencing, like CRISPRa. So look it up. If you're interested, come talk to me also. I didn't put any barcode. It's like, you won't find it. It's called CRISPRsec. And it analyzes both cases that I just described to you. It analyzes if we really want to target one region because we knocked in, knocked out, or modified one gene. And it also allows to analyze these screening experiments. Here you can see kind of doodles of plots that you can get out of the pipeline. Why did we decide to go for two workflows? Because a lot of scientists, and we found a lot of papers where they firstly do functional genomics, where they're trying to find good candidates. We also know that biologists tend to have favorite genes, so they also kind of look up like, oh, look, my favorite gene is in there. And then they proceed to also really go into a more tailored expression, tailored experiment, and then do like one region per sample. So this is why we decided to go for that. This is the workflow. I am not a graphical designer, but and I have worked during the hackathon on improving the tube map, but I am still quite proud of my skills on Inkscape. On the top part, you will find more the case where we are targeting one region, and on the bottom part, the more functional genomics aspect. We also, sorry, it's a bit hard with it, okay. Okay, I'd like to draw your attention to here. This is really the core of the functional genomics part, where we are looking at how essential are the genes. We can see that we have different modules. Half of them are on the NF core modules and have been reused, which was really nice for me also to see that when you, like, community-driven, to see what you do being reused. And what we are working on is also when you use several of these modules that you have a consensus, which I'm gonna show now. So when you use CRISPRsec, you get these kind of plots that show you which genes have been noticed as being interesting. We call them essential genes, or we can also talk about synthetic lethality. And then you can really pick from these plots. We start from FASTQ files and already get results, biologically relevant results. When we use, for instance, here I'm using two modules that have the same intention. We also allow comparisons of when modules, we kind of detect which modules are being run, and we give the user a comparison, a silly little Venn diagram to also allow them to really be wide or very narrow in the different genes that they want to investigate. We also, adding an extra layer, which I find really, really neat, is we're adding a layer with the protein-protein interaction. A gene is maybe even more interesting when we know that they're interacting with a bunch of different proteins. So we have that information in the pipeline, and we give it a higher threshold and higher value when it's really interconnected. So there's a bit of edge graph coming along, and this is also thanks to NF Core, because I am one of those PhD students who develops a software, and having it on NF Core really gave it more visibility, and we were contacted also by the University of Dublin to collaborate together, so that was really, really nice to work on this feature. And then for the So let's say I pick a gene, I want to cure cancer, I'm going to really target that gene, and I want to make sure that my insertion or my knockout worked well. This is the result plots that you would get. You have to know if your CRISPR-Cas knockout actually worked. So we get these kind of pie charts that give us the percentage of wild type versus the percentage of indel. So if we are looking for a high percentage of indel, which would tell us that our knockout experiment worked. Here, for instance, we also see exactly the second plot, the insertion and the insertion size, and exactly on the gene where what am I inserting where, basically. Okay, quirky things about our pipeline. I think LLM and AIs are a little bit like the talk of the last 2 years. Well, we also wanted to have a look at that. We also saw that Paolo developed a plugin which we are playing around with. So this is the work of Leon Hornig, who's currently doing his bachelor thesis at Cubic. So he added a little feature that right now is still on the dev branch, where the user can put a prompt, and we automatically also feed it the genes that are deemed essential and relevant, and we kind of get several outputs. And the next part is we want to compare it to how does it look like on a few publications that we already have. So we want to kind of assess the quality, and for us it is also the first time that we start from FASTQ files and we have a complete downstream interpretation. The next step is like, is this interpretation actually good? And also with the development of different LLMs, we'll be able to kind of track the quality. And for now, if I had to criticize this right now, it's not like LLMs are not reproducible, but we do assume that they will be soon, or that different LLMs will pop. So right now this is a GPT LLM, and we are currently integrating it in the pipeline. Yes, with that I would like to thank Cubic, of course. So Julia, a special shout out, you're mentioned twice, I also noticed. And I would like to thank Cubic, the NF Core community, and the Hattie Institute. Thanks a lot.